This method can help answer questions regarding differentiation of pluripotent stem cells in the retina field, such as the generation of different retinal cell types from human-induced pluripotent stem cell or iPS cells. The main advantage of this technique is that adherent human iPS cells can be directly used for the generation of retinal organoids, bypassing embryoid formation only by successive change of culture media. Laboratory engineer Amélie Slembrouck-Brec will be demonstrating the method to generate and isolate retinal organoids, and laboratory engineer Céline Nanteau will be demonstrating the strategy for retinal pigmented epithelial or RPE cell production.
Start the iPS cell differentiation following this schematic, which can be found in the text protocol, when the colonies reach 60 to 70% confluence. Prepare 4 milliliters of basal iPS medium per 6 cm dish and warm the medium to 37 degrees Celsius. Then, change the iPS cell medium to basal iPS medium. Note this time as day 0. At day 2, switch the cultures in the basal iPS medium to a basal iPS N2 medium, previously warmed at 37 degrees Celsius. Change the medium every 2 to 3 days. Identify emergent self-forming retinal organoids by neuroepithelium buds. At day 28, prepare 6 well plates containing 4 milliliters per well of